So let's now discuss the functional magnetic resonance imaging. Perhaps this is the most popular method in neuroeconomics. This is a quite complex method. That's why I strongly recommend you to read some specialized books describing the details of the functional magnetic resonance imaging. For example, this book written by Scott Huttel and colleagues. So today I will only focus on the major aspects of the MRI or functional MRI methods that will help you to understand the results of neuroeconomic studies. Well, briefly speaking, when we will put a subject inside of the magnet, his head will be surrounded by a few different coils. So first coil, a magnet, produces a very strong magnetic field. So why do we need the strong magnetic field? Because protons of the brain will be aligned in line with the strong magnetic field, and field is extremely strong. So if you will put a metallic coin in front of the scanner, it will fly inside of the scanner. So next, we can find a gradient coil inside of the scanner. This gradient coil produces a slight gradient in the magnetic field that will help to detect the position of the activation inside of the brain. And finally, there is a radio frequency coil that produces a radio frequency magnetic field that excitate protons during the scanning. So let's now discuss the basic facts behind this technique. So first of all, when you put the subject inside of the fMRI scanner, the protons are aligned in line with the strong magnetic field. Next, you apply to the brain the excitation radio frequency magnetic field. And this magnetic field activates protons. When you switch off this magnetic field, you can record the magnetic field emitted by the activated protons. This emitted magnetic field will be slightly different depending on the tissue that surrounds the protons. So using special algorithms, we can reconstruct the signal produced by gray and white matter or produced by active brain regions or less active brain regions. So here we use a quite complicated device, and I strongly recommend you to read some literature that would explain you the details of the fMRI method. So as you can see in this picture, MRI device is a very strange environment for their neuroeconomic studies. We use special projectors to project stimuli inside of the scanner. We use special devices to record behavioral responses of the subjects. We avoid any metal inside of these devices to avoid an interference with the magnetic field. But we are able to record brain activity and also record the brain structure of our subjects during the specific behavioral task, during the decision-making process. So there are two major types of the MRI studies. The first type is called the structural MRI. So with this method, with this protocol, we can collect the 3D atlas of the brain of our subject. But we also can use another protocol called functional MRI. With this protocol, with this sequence of pulses, we can collect the activations of different brain areas during the task. So using the second approach, so-called functional MRI, we can record the brain activity during the decision-making process. So in this case, we put our subject inside of the scanner. We put sometimes, for example, a small screen in front of his face, and we ask subject to make decisions inside of the scanner. So we record decisions during the scanning process, and we also record the brain activity during the decision-making task. The result of the neuroeconomic study often looks like this picture. So you see an activation inside of the brain. So what does this activation mean? The fMRI method can be explained by the very interesting study conducted 100 years ago by Angelo Mosso. He put his subject on the scale and asked subject to perform active mental calculations. As a result, scale turned. So why do you think that happened? Basically, when we make some calculations. Our brain is activated, so neurons need more energy, they need more oxygen. It triggers an inflow of blood to the brain, and it changes the weight of the brain. So this experiment actually explains the principle of the fMRI method. Our brain is penetrated by blood vessels. So these blood vessels bring oxygen to neurons. And whenever our neurons are active, it triggers an inflow of blood, an inflow of oxygenated hemoglobin, an inflow of energy to this region. And this inflow of oxygenated hemoglobin changes the magnetic properties of the tissue and can be detected by the magnetic resonance imaging scanner. So overall, functional magnetic resonance imaging detects the BOLT signal, blood oxygen level dependent signal. So when our neurons are active, when they produce a lot of action potentials, they need a lot of energy. So activity of the neurons triggers an inflow of blood to this region, an inflow of oxygenated hemoglobin. And oxygenated hemoglobin changes the magnetic property of the tissue and can be detected as an MR signal by the scanner. So with functional MRI method, we record an inflow of oxygenated hemoglobin to the active regions in the brain. So this picture illustrates that the result of the study normally is a combination of two methods. With structural MRI, we can collect the anatomical information about the brain of our subject. It takes normally 8-15 minutes, and you collect the 3D atlas of the brain of your subject. And next, you ask person to perform a task. And during this task, you record functional data using functional fMRI. So next, you analyze your data and you visualize it as the result of the statistical analysis. So this yellow color indicates a significant change in the activity of the brain region triggered by the certain task. So actually, this activation has nothing to do with real activity of the brain. This is a result of the statistical test. And the lighter the color is, the more significant is the modulation of activity in this brain region by the task. So once again, functional MRI is sensitive for BOLT signal. So it actually captures the change in the concentration of the oxygenated hemoglobin in the brain. Since oxygenated hemoglobin is diamagnetic, it actually increases the MR signal from the brain, and that can be detected by the MR scanner. So overall, neuronal activity triggers the local blood flow and an increase in the concentration of the oxygenated hemoglobin. It changes the magnetic properties of the region that can be detected as a large MR signal by the scanner. So with the functional MRI method, we detect the activity of the brain during the task. Basically, what do we see? The inflow of oxygenated hemoglobin to the brain that correlates with the activity of the neurons. So this picture illustrates the signal detected by the functional MRI method. The inflow of oxygenated hemoglobin to the brain is a very small process. So it takes a few seconds before oxygenated hemoglobin will arrive to this active brain region. So that's why MRI scanner actually is sensitive to the very slow change in the brain activity, and it is not sensitive to very fast changes in the decision-making process. It has relatively bad temporal resolution. So if I will overlay the BOLT signal, the MRI signal, and the actual neuronal activity, you will see that neuronal activity takes place first, and with some delay it triggers an inflow of oxygenated hemoglobin to this region. So actually MRI signal is delayed relatively to the neuronal activity. So with MRI, with functional MRI, we record the inflow of oxygenated hemoglobin to the region. So we record the neuronal activity indirectly. This MRI signal is proportional to the neuronal activity, but this is not direct measure of the neuronal activity. So once again, with functional MRI technique, we record some maps characterizing the inflow of oxygenated hemoglobin to the brain. We can statistically analyze these maps and find their changes in the activations of the brain related to the task. So normally, these results are overlaid with the structural MRI images. So what do you normally see in the neuroeconomics papers? This kind of statistical maps. And color here indicates the level of the significant effect. How significant is the change of the activity in the certain task. So whenever you see this kind of picture in the neuroeconomics paper or in media, you do not see the actual activity. You see the outcome of the statistical analysis. And color here indicates the level of statistical significance of the observed effect. So how do we get the statistical maps? We can simply record the brain activity in condition A, condition B, and subtract condition A from condition B. So we can see here on this graph the difference in the activation of the brain in condition A and B. So we can make the same calculation for many subjects and apply group statistics and show that this difference is reliable across different subjects. So the results of the statistical analysis can be presented differently. So it can be visualized as a slice of the brain, as a 2D image, where statistical map is overlaid with anatomical slice. It can be visualized as a 3D image. So the minimum unit here is the voxel. And voxel is similar to pixel in digital photos. But voxel is a 3D unit. So once again, whenever you see an activation of the brain, you see activated voxels, they represent the results of the statistical analysis. If you would like to see the actual activity of this region that is behind the statistics, you can visualize it using special programs. So for example, here I show you the bolt signal in this particular voxel. As you can see, the signal is stronger in condition 1 compared to condition 2. So this difference is represented in the statistical map as a result of the statistical analysis. So sometimes during this course you will see statistical maps. Please remember that this is the outcome of the statistical analysis. Sometimes you will see the actual activations of these regions, both responses. So here you see the time course of the activity inside of this region. And you clearly see that in the condition 1, this activity is stronger than in the condition 2. So the take-home message can be relatively short today. So whenever you see these statistical maps, these activations during the certain tasks, please remember that color here indicates the level of significance of the effects. So how reliable their activity in this region is modulated by certain aspects of decision making. So this result is a combination of the functional MRI and structural MRI. So you can collect structural and functional data during separate sessions. So it takes 7-12 minutes to collect structural data. And it can take you from 10 to 40 minutes to collect functional data. So you analyze the functional data and you visualize the results of the analysis, the results of the statistical analysis as a statistical map as presented at the bottom of the slide. So the results of the neuroeconomic studies can be presented very differently. Traditionally, in neuroeconomics papers and also in cognitive neuroscience papers, you will see 3 major ways of presenting the outcomes of the fMRI studies. You will see coronal, sagittal, and horizontal slices. So don't be surprised by different slices presented in the neuroeconomics papers. So for example, here you see the sagittal and the coronal slice of the brain. So each cluster each statistical cluster in the brain is called the activation. So normally, these pictures are supplemented by the tables. So in the table, you can find the name of this brain region, you can find the coordinates of this region. You also can find the size of this activation, the number of voxels in these activations, and also the level of the significance of this activation. So don't be surprised when you will see this kind of picture. So on the left, you see the statistical map. On the right, you see the actual activation of this region. So here, we present to you the activity of the ventral striatum that is related to the expected magnitude of the monetary outcome. So the statistical map shows us that activity in this region is significantly modulated by the expected monetary outcome, by the magnitudes of this outcome. On the right, you actually see the activity inside of this region, and you clearly see that this activity is particularly strong when person expects to get $5. It is less active when person expects to get $1. And actually, this region is deactivated when person expects to get $0. So don't be stressed by these pictures, and I hope that now you will be able to read these neuroeconomics results. So overall, fMRI method is most popular method in neuroeconomics. It is non-invasive, so we do not damage the brain during the scanning. It has a relatively good 3D resolution. Unfortunately, this is indirect method, so we do not measure directly the neuronal activity. We measure the bolt signal. We measure the inflow of oxygenated hemoglobin to these regions. It is quite noisy. It produces a lot of acoustic noise, and some groups of subjects are restricted from these studies. For example, pregnant people are not allowed to be scanned. So this graph represents the main neuroscience methods used in neuroeconomics. We use two axes here. So one axis represents the spatial resolution, the second one represents the temporal resolution. As you can see, fMRI method has relatively bad temporal resolution, but relatively good spatial resolution. MEG and ERP methods have quite nice temporal resolution, but very bad spatial resolution. That's why we have to combine different methods to characterize different aspects of decision making. If we would like to use MEG or EEG, we can study the dynamic of the decision making process. If we would like to localize the decision making process inside of the brain, perhaps we have to use fMRI methods and other methods. So normally we combine different methods to understand different aspects of decision making. Unfortunately, normally we have to repeat the task many times. We cannot study the single decision. To extract, to analyze the brain activity during the decision making process, we have to repeat the same decision many times. So we need quite complex statistics. Normally we report the group data, so we report the results for 20, 25, 30 subjects. Unfortunately, these techniques require laboratory experiment. That's why we cannot sometimes conduct studies of real-life decisions. Hopefully, after this lecture, you will be able to read the results of the neuroeconomic studies. You will be able to understand pictures like this. Next week, we will start our journey into neuroeconomics. We will discuss the major concept of neuroeconomics.